lectures on food microbiology for the fourth stage students of biology department in the College of Science in the University of Baghdad by Assistant Professor Dr. Marwa al-Khafaji, a microbiologist in food poisoning. In our third lecture, we will study the microbial contamination sources of food. First, we may define two important abbreviations, which are FFF, from farm to fork, and FFTT, from farm to table. There are two sources of food contamination, either natural sources or food contamination during handling and processing. Regarding natural sources, they are either the water or the soil, manure and sewage, animals and plants, and the air. Water is in continuous contact with foods from field of production or from farm to table. It may be divided to irrigation water, cleaning water, processing water, and cooling water. Water of irrigation in the river is very high contaminated by microorganisms. It looks like a culture media. It may contaminate food by two groups of microorganisms. These are psychrophilic normal bacterial flora of the river and mesophilic contaminating flora. The first group, psychrophilic normal bacterial flora of the water, is responsible for food spoilage in a refrigerator, while mesophilic contaminating flora is responsible for foodborne disease and food poisonings. So the psychrophilic normal bacterial flora of the river responsible for food spoilage in refrigerator because they are cold loving bacteria, while the mesophilic contaminating flora come from sewage or from wastewater, responsible for food-borne diseases or food poisonings. Soil, manure, and sewage. Soil is rich in microbial spores and parasite cysts, bacterial and fungal spores, especially those heat-resistant spores that resist heat treatment of the food. Sewage and animal manure used as fertilizers contaminate food with very dangerous bacteria, virus, and parasites. The third natural source of contamination of the food is the animals and the plants. Plants, particularly leafy vegetables loaded with microorganisms, because with continuous contact of the source of contamination, like soil, sewage, water, manure, rodents, these microorganisms reach the food produced from raw plants. Generally, animal origin foods more dangerous than plant origin foods because of two important reasons. Number one, there are 100 microbial diseases associated between human and animals. It's transmitted to human by food and products from these animals, like Creutzfeldt-Jakob disease or mad cow disease and other diseases. The second reason that why animal origin foods more dangerous than plant origin foods is the microbial contamination of animal origin foods, milk, egg, meat, produce toxic chemicals to human, like toxic polypeptide, fatty acids, ketones, and aldehyde. The fourth source of food contamination is the air. But we noticed that air in open field production is poor in microorganisms due to four reasons. Number one, air is poor in organic matter. Number two, low moisture because of the sun heat. Number three, bactericidal effect of the sun UV light. And number four, continuous removing of microorganisms by wind current. Our real problem regarding air source of contamination is the air of closed food plants, restaurants, food stores, because it loaded with microorganisms may serve as a source of contamination of food and the prepared meals. So it is necessary to disinfect air of closed buildings or restaurants or factories of the food using bactericidal chemicals or UV lamps or using a special cooling system, other sanitary practices, like replace oxygen by nitrogen in food stores. The second source of food contamination is the food contamination during handling and processing. In addition to the natural sources of contamination, the food exposed to heavy contamination during a long way from farm to table through 4 stages. The first stage, harvesting or collection of raw food in the farm. The second stage, during transportation. Third stage, manufacturing in the food plant. Fourth stage is marketing and distribution. During the first stage of harvesting or collection of raw foods in the farm, the food may be contaminated by soil, rodents, manure, birds, collectors, boxes. During the second stage, transportation, food may be contaminated by boxes, containers, collectors, or the trucks, where in the truck there's a special issue because food may become contaminated from the boxes in the cars that form a permanent society of contamination, which formed from food residues in these boxes or containers in the car. So don't forget permanent society of contamination related to trucks during the second stage of transportation. There is contamination during the third and fourth stages during handling and processing and marketing and distribution. But the most important stage participating in adding contaminant to food is the processing of the food, especially the factory or food plant. In the food plant, food exposed to all sources of contamination: air, water, soil, sewage, rodents, insects, food handlers, surfaces, equipments, containers. So the manager holds a big responsibility for production of food with the following characteristics: clean food, pure food, safe food, food with normal character, food with high nutritive value. In order to achieve these goals, there are many quality control sections established in each food plant. Number 1, pest control section. Number 2, chemical control section. Number 3, cleaning and sanitizing control section. Number 4, microbiological control section. Number 5 is the food handlers, employees, workers, health section. Microbiology quality control food plant. In the sites and materials of food plant. Number 1, the microbiology of processing water and drinking water. In this section, in addition to pathogenic microorganisms, we should examine slime-producing bacteria, iron and manganese bacteria, undesirable bacteria. In addition to microorganisms, chemical composition of the water must be examined, like the hardness of the water, iron and manganese content, and excessive organic matter. The second section is the microbiology of products. We must examine raw materials, ingredients, food additives, and product packaging material. In the microbiology of sewage and wastewater treatment unit, we must examine it. The microbiology of food plant equipments, from the floor to the ceiling, walls, windows of the food plant, doors, sinks, air processing surfaces, and equipments. In the microbiology of food stores, carton, bag, container, cars, trucks, etc. Microbial examinations of food handlers, workers, employees. The food handlers are very dangerous source of food contamination because they in contact with food from farm to fork. So they may contaminate food and prepared meals with two groups of pathogenic bacteria or microorganisms. Number 1, respiratory system pathogens through sneezing, coughing, bluffing, chatting by droplets. Number 2, enteric pathogens due to improper hygiene habits. Food handlers' external feature must be similar to that of a heart surgeon. Why? Because if he contaminates the food, hundreds of consumers may be killed, while the surgeon may kill only one person, the patient in this case. We see this careless food handler while this careful food handler. These are a list of the main microbiological tests on food plant. TBC, total bacterial count. TYMC, total yeast mold count. TPBC, total psychrophilic bacterial count. TTBC, total thermophilic bacterial count. TSC, total spore count. TCC, total coliform count and fecal E. coli. Enterococci test, Clostridium perfringens test, test for pathogenic bacteria, test for parasites, test for viruses, test for bacterial toxins, and test for mycotoxins. We will talk about food adulteration. You see this food? What about the donkey? The famous worldwide organization Food and Drug Administration (FDA) consider the food product and food plant as an adulterated food if one of the following cases occurred. Number 1, if the food contains faulty, putrid, or decomposed substances. Number 2, or the rule number 2, if the food contains poisonous, unsafe chemicals or food additives. The rule number 3, if the food has been prepared, handled or packed under unsanitary conditions, whereby it became contaminated with microorganisms and chemicals. The rule number 4, if the food product is produced by a diseased animal or dead animal, not by slaughtering. The rule number 5, if the food container is composed of a poisonous substance. Thank you for listening. I'm available for any question.